Hello everyone, myself Deepa Singh, working as assistant professor in Gyan School of Pharmacy, SGVU Jaipur. Today we discuss about drug clearance. In previous lecture, we already covered renal and non-renal drug of excretion, means both excretion route, including renal and non-renal. Today we discuss about drug clearance, how drug clearances takes place, different method, how we calculate this drug clearance, and how we calculate or different method for calculating this drug clearance. Including this, we covered small introduction of bioequivalence or viability because it's the next topic. So we just include a small introduction part of this viability, and before this we know complete information regarding drug clearance. It's a topic of biopharmaceutics and pharmacokinetics, 6th semester biopharm subjects. So before starting this topic, you should know about basic object or learning objects. In learning object, introduction of the drug clearance of the drug means what is a drug clearance, the introduction or brief description about the drug clearance. All this know about this topic and how to calculate this drug clearance, different method to calculate the drug clearance, or how you use this method to calculate the drug clearance and clearance model. There's a different models available for this drug clearance calculation. So you know about all this model or how you use this model and introduction to viability. All these are the basic object and introduction part just to cover what is viability and difference with viability and bioequivalence. And you know complete detailed description about the drug clearance. And in this introduction part, calculation methods, all these we covered in this topic. In this, drug clearance is a pharmacokinetic term for the describing a drug elimination from the body without identify the mechanism of the process. Means drug clearance is a pharmacokinetic process. We already discussed that pharmacokinetics include AD, ME, absorption, distribution, metabolism, and excretion. So this pharmacokinetics parameter is also include this drug clearance. Drug clearance is a pharmacokinetic parameter and it describes the drug elimination from your body. Means with the help of this drug clearance, you calculate the elimination rate or how much drug is eliminated out in your body. And we already discussed that the elimination include both the metabolism and excretion. So with the help of this drug clearance, we have to calculate the elimination rate from the body or how much drug is eliminated from the body or without identify the mechanism of the process. And in this, instead of describing the drug elimination rate in the terms of amount removed per unit time, it's a unit that you calculate how much drug is clear or how much drug clearance takes place in your body. And after the calculating, this is a formula that is mg per minute, how much mg drug excrete out or eliminate out to your body in 1 minute or per minute. So mg per minute is the formula unit of that particular method. And in this drug clearance is defined as a fixed volume of the fluid. Means all fluid are included in drug clearance. In this containing the drug or maybe any other drug cleared for the drug per unit time. And means this drug clearance is you calculate by the how much fluid volume is excreted out or how this fluid volume containing the drug in per unit time or how much time is taken to complete removal. And the unit for clearance are the volume per time, that is ml per minute or liter per hour. It's a drug that is cleared through in your body in mg per minute, means how much drug is cleared in mg in 1 minute or how many minutes mg per minute. And it's a fluid volume or volume that cleared through your body in ml and liter in maybe minute or in an hour. That is a volume unit of the clearance volume per time. In time, maybe it's in a minute or maybe in an hour, or if you are taking in a minute, so you consider maybe in ml because it's a small volume. And in liter, you consider as an hour, means how much liter is excreted out of clearance out in particular 1 hour or 2 hour. For example, if the clearance of penicillin is 15 ml per minute in a patient and penicillin has a volume of distribution of 12 liter. In this, we are taking one example of penicillin. In penicillin, 15 ml of clearance takes place in 1 ml, or in 1 ml, 15 ml clearance takes place, and the penicillin volume of distribution is normally 12 liter in your body. And from the clearance definition, 15 ml of the 12 liter will be cleared from drug per unit time. So total is 15 ml, and 12 ml is cleared in 1 minute. And it's a volume of distribution as 12 ml liter. So it's an example of penicillin. In this alternative clearance, total clearance may be defined as the rate of drug elimination divided by plasma drug concentration. Means it's a formula for clearance is that a ratio of how much drug is eliminated or how much drug is excreted out to the body, and you divide this ratio by plasma drug concentration. Means you can say that clearance is a ratio of drug present in your plasma or in your body, and that is eliminated out divided by plasma drug concentration, total drug present in your plasma. So formula, clearance is equals to elimination rate divided by plasma drug concentration. Clearance is VDE divided by DT divided by CT, and unit is ml per minute, means how much ml is excreted out in 1 minute or maybe particular that minute. And just as the elimination rate constant K represent the sum of total of all rate constant for the drug elimination. Means this elimination rate constant K or K is represent the total amount of drug is cleared or eliminated out to your body, or some of the total rate constant of the elimination, including excretion and biotransformation. Clearance is the sum of the total clearance process. Means it is also, you can say that is sum of the total clearance process in your body, including clearance to liver, kidney, lungs. And you can say that when drug is cleared from the kidney, then it's known as renal clearance. And when drug is excrete or eliminate out from lungs and liver, then it's called as hepatic clearance. So it, and also some of this clearance. So the example, simplest case beaker method. In the simplest case beaker, you can say this is a beaker, and rather than describe the terms of amount of a drug removed per unit time, clearance is described the volume of plasma cleared per unit volume. Means you can say that it's a unit volume per time. Volume, suppose you can take maybe mg or in ml or in a liter, and time maybe in a second, minute or in an hour. So it's a formula. You take this formula or modify this formula according to your clearance rate, maybe mg, ml or liter, or time is maybe minute, seconds or in an hour. And suppose this is a 100 mg, ml, liter, and it exceeds out consider as a liter in per hour, and total is 10 liter. And this excretion or elimination rate is 100 mg per liter. And in this, there is 1000 mg per drug present in the liter. So it's a simple beaker case method that you calculate how much drug is removed or eliminated out to your body in a particular unit or particular time. But the body does body not a beaker, multiple system involved. And in this there's a diagram showing both renal and hepatic clearance. Drug is entered and it goes in your kidney and liver, and when this is a complete volume of distribution after the kidney, then excrete out by urine, that is KE. We already discussed this excretion in the renal excretion part. And if drug is excreted out by liver or through bile and metabolized, this is Km by metabolized, and K bile is the drug is excreted out by bile. Means there may be chances that drug is excreted out by kidney or liver or by the both route. And out of total volume distribution, calculate how much drug is excreted out through that particular site. This clearance model. In clearance model, the calculation of a clearance from a K and volume of distribution assume that sometimes incorrectly. Means you don't calculate properly this constant clearance rate and volume of distribution. You just assume, and due to this assumption, it shows an incorrect rating or incorrect procedure. So in this, a defined model are formed, and use this defined model, and the clearance estimated directly from the plasma drug concentration time term does not assume any model. And for calculating this clearance model, we can assume that plasma drug concentration model or curve of plasma drug concentration. With the help of this plasma drug concentration, we do not assume any model, and we can calculate this value with the help of drug concentration time curve. And in this physiological organ clearance and physiological organ clearance may be calculated for any oxygen involved in the irreversible removal. Means in physiological organ, you can say that this physiological organ clearance may be calculated by any oxygen involved in irreversible removal of a drug from your body. means how much drug is removed irreversibly from your body, or how much oxygen involved for removing or clearance of the drug through your body. And many organs in your body have a capacity for the drug eliminated, including drug excretion and biotransformation. Means you can say that this elimination takes place in many body and according to the capacity of the drug in your body, and this elimination include both excretion as well as metabolism, or maybe biotransformation. Metabolism and biotransformation both are similar, or the synonym of biotransformation is metabolism. So in elimination, we include both excretion as well as biotransformation rate. And rate of elimination by organ is equals to rate of presentation organ input minus rate of extraction organ output. It is QC in minus QC out. So you calculate drug clearance, means how much drug is input or in that particular organ inside, or rate of presentation organ input minus rate of excretion organ output, means input minus output, that is QC in minus QC out. Physiological organ clearance, it's a formula. And in next clearance, that is Q, and we will calculate this. Q is if the drug concentration in the close or in a blood, that is Ca. Concentration in the blood means Ca, and entering is greater than the drug concentration of the blood, that is Cv, leaving the organ. Then some of the drug has been extracted by organ, and the ER is Ca minus Cv divided by entering drug concentration. Means you can calculate clearance rate by this formula. Clearance is Q, or you can say that it's a ER. In this, we calculate drug is enter in your blood or drug concentration in blood, and after that drug how much is leaving that particular organ, or concentration of drug to leaving that particular organ. And Ca is means entering of a blood or drug present in your blood, and after Cv is means removal or leaving of a blood from that particular organ. So you minus Ca minus Cv for clearance divided by Ca, means how much total drug entered that particular site, and it's a formula. And for hepatic clearance, we can say that clearance is H, because we are calculating hepatic clearance. That's why we write down CLH, clearance for hepatic. And it's again similar to the previous one. In this, you calculate how much drug is eliminated through liver, because we are calculating hepatic clearance. So we are taking just individual organ or particular organ which we are calculating. So here we are calculating clearance rate in hepatic site. So clearance in hepatic site is the or rate of eliminated by liver. How much drug is eliminated by that particular liver site, and how much drug total present in plasma concentration. So it's a formula: rate of elimination by liver divided by plasma concentration. And clearance in hepatic is equal to clearance in total minus clearance in middle. Next is model independent method. In model independent method, clearance is commonly used to describe first drug elimination from the compartment model, such as one compartment model. We discuss this one compartment and multi-compartment model in the next lecture or maybe in the next unit. And in this model independent method, mainly we consider as one compartment model. You use first order kinetics elimination rate for describe this compartment model, such as one compartment model. And model independent method are non-compartment model approach. It's a non-compartment model approach, or non-compartmental method used to calculate a certain pharmacokinetic parameter. In this certain pharmacokinetic parameter, you can calculate clearance or viability both, and viability denoted by F. And this independent method is a model independent method, and with the help of this clearance rate and viability, calculate the pharmacokinetic parameter and the clearance rate. And major advantage of model independent method is that no assumption for a specific compartment. You don't assume or don't take any assumption for that particular specific compartment or any other compartment is required to analyze the data. And whatever the volume of distribution and elimination rate constant need to be determined. Means in model independent method, you just calculate or determine the volume of distribution and how much drug is distributed out, and elimination rate constant. How much drug is eliminated particular rate constant. So this is about model independent method. Next is compartmental method. In compartmental method, static volume and first order elimination is assumed. Plasma flow is considered as a clearance is equals to KVD. In compartment model, it's just opposite to independent compartment model. In this, we can take or assume first order elimination constant rate or first order reaction, and plasma flow is not considered in this compartment model, and you calculate the clearance. In fact, this formula clearance is equals to constant, that is K into VD, and in physiological model, clearance is the product of plasma flow Q and extraction ratio, that is ER, and clearance is equals to Q into ER. It's a formula for physiological model and it's a formula for compartmental model. And in this compartmental model, clearance you calculate Q into ER, and in compartmental model you calculate clearance is equal to K into VD. But in physiological, you multiply Q into ER, and in model independent method, volume and elimination rate are not defined. In model independent, you just check or assume, and in this no specific volume or a specific rate of volume and elimination is determined. It's not a defined amount. It may be vary in different range. And in renal clearance is defined as the volume of plasma drug is cleared of the drug per unit time through the kidney. Means you can say that renal clearance is the amount of a drug or amount or volume of drug present in plasma that cleared through kidney or through specific renal route, and unit maybe select according to drug elimination rate per unit time through that particular kidney. And similar renal clearance may be defined as a constant fraction of the volume of distribution. Means you can say that this renal clearance is also a constant friction of volume of distribution. The drug contained that is excreted out by kidney per unit time, how much drug is excreted out by kidney at particular time. And more simply, renal clearance is defined as urinary excretion rate, that is PDU divided by DT, and then divided by plasma drug concentration. In simple way, we already discussed the renal route of excretion. So this renal clearance is the amount of a drug that is excreted out by this renal route or by kidney in a particular unit per time or a specific volume. So you can calculate this renal clearance, and when we can say that the formula for renal clearance is also very simple, and you just divide how much drug is eliminated or how much rate of drug elimination takes place by kidney, and divide by plasma drug concentration, means how much total drug present in the plasma. So it's a ratio of rate of eliminated by kidney divided by plasma concentration. The formula for renal clearance, and you denote by D2 divided by D2. This is the concentration or elimination rate in kidney divided by CP, plasma concentration. Rate of drug passing through kidney is equal to rate of drug excreted, means how much drug is passed to that particular site or to that renal route, or after passing how much drug is excreted out, you can calculate. In this, clearance is equals to CP is equals to QU into CU. And when we can say that unit of this formula is clearance in ml, CP is in minute, and into mg per ml is equals to ml per minute into mg into ml. So that's regarding the formula of this clearance rate in kidney, and this clearance formula is ml per minute, and CP formula is mg per ml, and then QU formula is ml per minute, and CU formula is unit is mg per ml. It's a unit for this formula. This is regarding the drug clearance to this kidney or hepatic renal clearance. Next is comparison of a drug extraction method. In this, renal clearance may be measured without regard to physiological mechanism involved in this process. In this, you just compare total clearance rate or total drug that is secreted out of filter or maybe reabsorbed that particular site. When you calculate individual parameters, so you can merge or compile the total clearance rate or easily calculate that total clearance rate. In this comparison of extraction method, in this, renal clearance may be measured without regards to physiological mechanism involved in this process. No physiological mechanism is involved in this. You just calculate the volume and capacity of that particular site or particular mechanism. And physiological viewpoint, however, renal clearance may be considered as a ratio of some of the glandular filtration. Means you can say that clearance is maybe sum of, or maybe take a ratio of glomerular filtration and active secretion. That is, takes place by renal side or by the kidney, right? And less a reabsorption rate divided by plasma drug concentration. So it's a formula to drug excretion calculation. Clearance is equal to filtration rate plus secretion rate. Hence, you can sum the how much drug is excreted out, or maybe sum of granular filtration and plus active secretion. And after this sum, you can minus this reabsorption rate, how much drug is reabsorbed, or again showing that particular effect per particular site or ready to reabsorb, and divided by CP, means plasma concentration. And the actual renal clearance is not generally obtained by direct measurement, means you already discussed or already see that renal clearance. In renal clearance, we already divided different mechanism or different steps for this renal clearance. And in this renal clearance, you do not calculate directly. It requires certain formula or certain methods. And before calculating this clearance, it's a formula how much drug is filtered out through kidney, and secretion rate, then sum of this secretion rate and filter rate minus reabsorption rate, means how much drug is reabsorbed or again available for the absorption, and divided by CP, total plasma concentration or concentration of the drug in plasma. Next is filtration rate. In this filtration, we will calculate if glomerular filtration is solved process, the drug elimination is no drug is reabsorbed. Means you can say that drug excretion is takes place, and this no drug is reabsorption is take place in filtration only. And we calculate just how much drug is filtered out, and no data is required for reabsorption. And the amount of drug filtered at the any time T is means the amount of how much drug is filtered at particular time will always be CP into GFR, glomerular filtration rate. Likewise, if clearance of a drug by glomerular filtration only, as in case of inulin, then clearance is equals to GFR. Otherwise, clearance represent all process by which drug is cleared to kidney, including any combination of filtration, reabsorption, and active secretion. Means in filtration, you don't say that you use a reabsorption, you just calculate how much drug is filtered out at particular time and particular site, or through this concentration plasma or the filtration rate, and including the combination of filtration, reabsorption, active secretion. In this, ddu divided by dt is equals to Ka. Ke into Vd into Cp. It's a compartmental model formula, and in physiological method formula, we can say that du divided by dt is equals to clearance Cp, that is physiological formula. From the above equation, when we simplify that, you can write that Ke into Vd into Cp is equals to clearance into Cp, and Ka is equals to clearance divided by Vd. It's the equation of final clearance or final filtration rate or elimination rate. Ka is equals to clearance divided by volume of distribution. In this, you can say that we just write down this first compartment model, and second is physiological model. When we write down this equation, is equals to, and when you simplify the equation, you get this formula. K is equals to clearance divided by Vd. Clearance divided by this Vd, and Cp are cancelled. So K is equals to clearance divided by Vd. Next is model independent method. In model independent method, clearance rate may be also estimated by single non-graphical calculation from the knowledge of AUG, area under graph, 0 to infinity, the total amount of drug absorbed, and FD0, and the total amount of a drug excreted in urine, and du to infinity. And for example, a single IV oral drug injection is given to a patient, and AUG to 0 to infinity is obtained from the plasma drug line term form. Total body clearance is estimated by clearance. Clearance is equals to du, and divided by AUG, 0 to infinity means how much drug is eliminated out by this particular model at that particular site, calculate till infinity. And that's why I consider 0 to infinity, and clearance is equals to du divided by AUG, 0 to infinity. And if the total amount of a drug excreted in the urine, du to infinity, has been obtained by renal clearance, is calculated by d to 0 to infinity divided by AUG, 0 to infinity. And clearance can also be calculated from a fitted parameter if the volume of distribution and elimination rate constant are known as body clearance. Means this calculation part of clearance you also calculate, and in this mainly you can say that volume of distribution and elimination rate are known amount of known volume, and body clearance, renal clearance. Renal clearance is R, and total body clearance is notified or mentioned that P, and hepatic clearance is CLH and can be calculated according to the following expression. And this total clearance means total amount of drug is exuded or total drug present in your body. As clearance total is equals to KVD, means total volume of distribution in your body, and clearance in renal or by the kidney route, that is KEVD, elimination through this renal route, and clearance by hepatic route is calculated by KM into VD. So total body clearance is equal to sum of renal clearance and hepatic clearance, and based on the concept that entire body enters a drug. So you can also say that total body clearance is the sum of drug is excreted out or eliminated out through renal or hepatic as well as route, both type of the route. So when you compile this equation or calculate the total clearance in your body, so in this write down clearance is equals to clearance in renal route and clearance in through hepatic route, and sum of renal plus hepatic route, and VD cancelled. And you can also say that in simple way, when we calculate total body clearance is equals to, it's a sum of renal clearance plus hepatic clearance. For substitution the equation 6.35 and 3.6 into the equation 6.8 and 3.5 and 3.6 when substitute this in 6.8, then we get KVD is equals to KEVD plus KMVD. It's after substitution of three equation or two equation in 6.38, 6.35 plus 6.3 substitute, and equation 6.38, then we get K is equals to KEVD plus KMVD, and dividing this by VD on both sides of the equation 6.39. So 6.39 equation, and divide this equation by this VD on both sides here and here. So after dividing this VD, we get a simple equation or easy equation, that is KE plus KM, KE plus KM, because when we divide this VD, so VD gets cancelled, as VD also cancelled, and K is constant is equals to KE plus KM, and total body clearance is equals to, it is established by dividing the rate of elimination by each organ with concentration of the drug present. Means you can calculate how much total drug clearance takes place or how much drug is cleared, total amount in your body. So in this, total clearance is equals to clearance in hepatic plus renal plus bile or remaining route. Remaining means maybe non-renal route or any other route. So total body clearance is not only depend on this three route or not only single renal route, but it's all route including renal as well as non-renal route. And that's why we write down plus and then infinity. So total clearance is equals to clearance in hepatic, clearance in renal, and clearance in bile or remaining any other renal as well non-renal route. This is the formula of total body clearance. Next is protein bound drug. In protein bound drug are not eliminated by glomerular filtration, and protein bound drug are usually excreted out by elimination. Following capacity limited kinetics, means the protein bound drug are not eliminated or removed by glomerular filtration. That's why we do not calculate this previous formula. And these protein bound drugs are show active secretion and following capacity limited kinetics. The determination of the clearance that are separated by two components result in hybrid clearance. This show hybrid clearance, and there is no simple way to overcome this problem. And clearance for protein bound drug is therefore calculated by following equation. Clearance is equals to rate of unbound drug excretion divided by concentration of unbound drug into plasma. This formula is for protein bound drug or those drug is not eliminated or not excreted out by glomerular filtration or by the renal excretion. So use this formula, and this will calculate, or we can say that the ratio of drug in unbound form. It's not in a bound form, it's an unbound form. Drug in excretion and divided by concentration on unbound drug in plasma. It's also unbound drug in plasma, and it's unbound drug in excretion rate. Plasma protein binding has very little effect on renal clearance of actively secreted drugs such as penicillin. This is regarding protein binding. Now, second topic, or just we include a small introduction about the viability because that's the next topic of our lecture, and we take this description about viability in next lecture. So you know about what is viability, definition, introduction part of this viability. In this viability is denoted by F and generally expressed as percentage. F percentage means viability is denoted by, or another name of this viability is F. For calculating this viability, use F symbol, and when we express in a percentage, then we put the percentage symbol on to this F, and it's an F percentage, and qualify the proportion of the drug which is absorbed and available to produce systemic effect. Means this is a F percentage required or available to show the or absorb the particular systemic effect or particular site. And in this, you can say that viability is a fundamental property of pharmaceutical product for a given route of administration. Viability is also a fundamental property of pharmaceutical product property when we given the drug by a specific route of administration or by specific route of administration, and how much drug is available after administration to a particular site. So it is also known as show that reproducible for all drug product intended to produce a systemic effect. Hence, how much drug is available to produce systemic effect or desired if pharmacodynamic effect or therapeutic effect. So it's a reproducible amount. And in this, therapeutic effectiveness of a drug depend upon the ability of a dosage form deliver the medicament to its site of action at a rate of amount sufficient to elicit the desired pharmacological response. Means you can say that this viability is also depend on therapeutic effectiveness, how much therapeutic effect is observed in that particular drug, and in this you can say that ability of dosage form deliver the medicament at particular site of action where the action is required, how much drug is absorbed or show or reaches to that particular site according to requirement, and the rate of amount of sufficient elicit to decide the pharmacological response, and this rate is sufficient or not for showing or producing the pharmacological response. Means you can say that this viability is also a therapeutic effectiveness of that particular drug or that particular site, whether effective action or therapeutic action is required, and you check that these drug is produced or not, that particular pharmacological response or not. And in this attribute of the dosage form is referred to the physiological ability, biological ability, or simply viability. These are the three attributes that are referred to this dosage form in this physiological, biological, and simple viability. And according to these three attributes, to calculate how much drug is available for showing the therapeutic effect. And for most drug, the pharmacological response can be related directly to plasma level. And how much drug is available in plasma. And so with the help of this plasma level concentration drug present in plasma, you can also calculate this viability. In definition of viability, viability is a term used to indicate the fractional extent to which dose of a drug reaches to its site of action or biological fluid from which drug has access to its site of action, according to Goodman-Gilman. Goodman-Gilman say that the definition of this viability, or how we define this viability, means it's a friction or extent, how much drug is extended or available friction to reach that particular site or that particular required site, and the biological fluid is showing the effect or showing the required to show the effect which access that particular site of action or showing that particular side effect. Means how much drug or how much friction is available to that required particular site for show the effect to that particular site according to Goodman-Gilman. And in the another definition, the term viability is defined as the rate and extent, means amount of absorption and change drug in its dosage according to Brahman and Jaiswal. According to this Brahman and Jaiswal book, this viability is defined as an amount, and this is mainly depend on the amount. Amount means rate and extent of that particular drug available for absorption in an unchanged form to its dosage form. This is in Brahman and Jaiswal. Next, in other definition, like drug or genetic drug or pharmaceutical equivalent, because these are interrelated or similar to viability, these definition are needed for understanding this viability. So you also know about this other definition, and brand name drug. Brand name drug is a drug marketed under the proprietary trademark protected name. Means you just approve some drug or come in a market in a particular name. This name of that drug is known as brand name, and these drugs are as a come in a marketed under a certain property or proprietary condition or trademark protected name. No one copy that particular drug or should have that trademark or that particular name. That trademark and name nobody use to formulate the drug, and this is a specific proprietary condition, and it's come in the market due to by some approval, and nobody have that right to copy that name and trademark. So this is known as brand name drug. In generic drug, a generic drug is seen as a brand name drug in dosage, safety, strength, and how it taken, quality, performance, and intended to boost. Means this generic drug is also seen to brand name drug. First, you know about what is branded drug and what is generic drug. Branded drug means those drugs which first come first time in market and taking the patent for 15 to 20 years, and at that particular time nobody copied that product. It's known as brand drug or brand name drug, and that drug should have a particular trademark or name, and after completing the patent, someone take that patent or by using that particular patent copy the product after completing the patent, and copy the product that is called as generic drug. And this generic drug is overall similar to a branded drug, and it's just a copy of a branded drug. So we can say that efficacy and strength and safety and quality performance all are same of this branded drug. Use also is same for this branded drug because it's a copy of a brand drug or brand name drug. And pharmaceutical equivalent and drug product are considered to be pharmaceutical equivalent if they contain the same active ingredient, have the same dosage form, and route of administration, identical strength and concentration. Means after completing the patent, two person are formulating two same drug, and the effect is also same. All the property are same. This is known as pharmaceutical equivalent because we can say that both drug are in a considered amount or equivalent amount are same, and active ingredient or particular drug is also same dosage form you prepare, maybe both are in tablet form or maybe in a syrup form, or the route of administration is also same. You can take both drug by oral route or by parenteral route, and identical strength means both are in the same volume or same amount. This is known as pharmaceutical equivalent. Means just both drugs are similar but manufacturers are different, and the property, oral formulation procedure all are same. So this is known as pharmaceutical equivalent. Brand name means a person taking one patent of that particular drug and comes in a market first time and nobody copied that particular drug to a specific patent time. That's known as brand name drug. Generic drug is just copy of a brand name drug after completing that patent, but all property, safety, and efficacy, quality all are seen to a brand name drug. And pharmaceutical equivalent means two drugs are similar. These drugs are maybe generic drug, and these drugs have nothing is changed, both route, strength, and effectiveness, ingredient all are same, but manufacturers are different. This is known as pharmaceutical equivalent. Next is pharmaceutical alternative. This alternative are the drug that contain the same active moiety but different chemical forms such as ester or salt of the active moiety, or they may differ from innovative product in dosage form or strength. Alternative means someone prepare that particular formulation and just change little amount of formulation or change alternative drug or maybe the any other form of that particular drug or moiety of that drug. In this known as pharmaceutical alternative formulation, alternative drug. In this you can say that it contains same active moiety or maybe different chemical form. First drug is in a normal form and second drug is in a product form or maybe ester form or maybe salt form. So this is known as alternative pharmaceutical alternative, but innovator may be differ, or the innovator are two different person. One may be first that take simple drug, or second is maybe take salt form of that drug. So innovator may be differ or maybe vary This is known as pharmaceutical alternative. Referred listed drug, that is RLD, is referred listed drug, is an approved by the product to which new generic version and are compared to show the bioequivalent. Reference listed drug book is the standard book. In this, mainly you print up or write down all the approved drugs which are available in a generic version or maybe compared to similar equivalent version. This is regarding reference listed book. If the size of dose is administered in a scene, then the viability of the drug will depend on three major factors. In this, pharmacological factor related to physical property of the drug, characteristic of the dosage form, patient-related factor and route of administration. Means the three drugs are in the same form or same viability. In this, consider to just distinguish or distinguish this three parameter. In this, physicochemical property vary or patient-related factor or route of administration may be changed. Within the parental route, intravenous injection of the drug results in 100% viability and the absorption process is bypassed. If you are taking a drug by parental route or by intravenous route of parental route, so in this condition you observe 100% viability because you are taking drug by intravenous injection, and absorption process is just bypassed by this intravenous. However, for regional stability and convenience, most drugs are administered orally. Due to certain problem or convenience and stability or convenient problem, you administer the drug orally. In such cases, the dose available to patient is called as a bioavailable dose, and if often less than administered dose. And the amount of drug reaches to systemic circulation is for absorption is called as systemic viability and simple viability. Means how much drug reaches in systemic circulation is known as systemic viability and viability. The term bioavailable fraction and refers to fraction of administered drug that enter a systemic circulation. So F is also noted down with the ratio of bioavailable dose divided by administered dose. How much drug is available in your body or total amount of drug that administered in your body. To extend optimal therapeutic action, active moiety should be delivered to its site of action and effective concentration to desired period. This is all about viability or introduction of the viability. After completing this viability, you should know about this outcome. Description of the clearance of the drug means complete information regarding the clearance of the drug, how the clearance takes place. We already covered this topic. And this renal clearance, hepatic clearance, vial clearance, or all clearance to include in total clearance. Formula of this clearance, how we calculate the clearance rate, or specific formula or method for calculating this clearance rate, or certain models that we covered in this PPT. So in learning outcome, you should know all complete information regarding the drug clearance, including different methods or model that you use for this drug clearance, and a small introduction regarding the viability, what is viability and how you calculate, and different definition for this viability. This is regarding the learning outcomes. After this reference part, these books are good book for this particular topic. Leon Chavez, Susanna, W. Brown, Edru, B.C. Bayu, Applied Biopharmaceutics and Pharmacokinetics. Second is B. Veshkant, Biopharmaceutics and Pharmacokinetics, second edition. Next is Brahmankar D.M. Jaiswal, SB Biopharmaceutics and Pharmacokinetics. Next is student P.L. Bosco and Law Viability, and its assessment GVT Pharmacol Therapeutics 27. Patient wise 455 to 466. Next book is Biopharmaceutics and Pharmacokinetics. Presented by D.M. Brahmankar, Sunil D. Jaiswal, a second edition, Valla Prakash, page number 315 to 363. This is book for this particular topic. All books are good and sufficient content is all books. Next is the question part, which is related to this viability or that total clearance rate or expiration rate. In first question, hydrophilic medicine agent has the following property. The option is low ability to penetrate through the cell membrane. Liquid base penetrate through membrane by means of endocytosis. C, easy combination through blood-brain barrier. D is reabsorption in the tubule. In second question, what does term viability means? Plasma protein binding degree of the substance. The option is commonality through blood-brain barrier. This is a fraction of unchanged drug reaching to systemic circulation. And D option is amount of substance in urine related to initial dose. In third question, the process, the reason determining viability are, the option is rheological parameter of the blood, is amount of substance obtained orally or collective of intake. C is intake of absorption, hepatic first pass effect. D option is glomerular filtration rate. Next fourth question is route of administration and passage of the drug through the liver is minimized by A oral route, B is transdermal route, C is rectal route, and D is duodenal route. And fifth question, reason of complicated penetration of the strong drug through blood-brain barrier. The option is meningitis. The option is high lipid solubility of the drug. C option is absence of a pores in the brain capillary endothelium. D option is high endocytosis degree in the brain capillary. And in next sixth question, the term biotransformation include. The option is accumulation of substance in fat tissue. B is binding of a drug with the plasma protein. C is accumulation of substance in the tissue. And D option is the physical, chemical, and biochemical alteration of a drug in the body. Next, in seventh question, half-life is time required to. A option is change the amount of a drug in plasma by half during the elimination. B option is metabolized half and introduced drug into active metabolites. C option is absorb half of introduced drug. The option of bind half of the introduced drug to plasma protein. And in eighth question, the elimination rate constant is defined as the following parameter. A option is rate of absorption. B is maximum concentration of the substance in the plasma. C option is the highest single dose. D option is the half-life. Next is a ninth question. In ninth question, systemic clearance is related with. A option is only the concentration of the substance in plasma. B is only the elimination rate constant. C is the volume of distribution, half-life, and elimination rate constant. D is the viability and half-life. And last 10th question, in this the renal clearance of hemoglobin is used to measurement of. A option is effective renal blood flow. B is the rate of renal excretion. C option is active renal secretion, and D is a glomerular filtration rate. These are the four options. According to this, you calculate the renal clearance and how you measure this renal clearance of this hemoglobin. And we already discussed that renal clearance or renal excretion route as well as how renal clearance is calculated. So after completing this topic, you easily answer this question or you understand this question. So thank you so much for watching and listening this video. Thank you.